very easily relate to that. He had barely 100 houses in that village, and my village had 100 houses. He was born about 20 plus years before me, and I was about 6 years old when India got its independence. And I can tell you how poor India was. You know, I cannot find a word in English that can describe the poverty. Dirt poor. So that is where Govind starts. And he had another unfortunate hit. India got independence. There's a partition. His whole family literally had to leave everything behind, shattered lives, and they come to Delhi destitute. You cannot imagine that. And I had really very long association with Govind, other than maybe Tom. And it was a non-traditional association. I was never a postdoc or a researcher in Govind lab. I was at MIT due to Tom, and he introduced me to Irving, London, and that is, and it turns out that Irving, London, Tom Rajbhandari, and Professor Khurana, they were very good friends. And so I come back to the village, and I wrote those words for Govind, and those words represent the values that he picked up as a child growing up in the village. And the fruit never falls far away from the tree, and those values he picked up from his parents. And everybody has said all the nice things about Govind, and they represent the values that he picked out from the village in Punjab. Now, I am not going to, I want to keep time because there are other people who, yes. A lot of things Govind told me, and you have already heard about this guy, Gordon Shrum. I brought this picture, and one day he told me that I am extremely grateful to Gordon Shrum because not only he gave me a job, he encouraged me, he supported me. More importantly, he said he protected me. And while the last thing he said to me in this sentence, but that he was my second father. Next person I want to bring up, everybody has already talked about Esther, and what nobody knows, because I had this non-traditional relationship that I was never a postdoc, and I could interact with him at a somewhat more relaxed position environment, although also I had this medical degree which allowed me some measure of observing Govind from a different point of view, listening to him from a different point of view. And so it kind of gave me a little bit different perspective of Govind. Extremely nice, extremely nice, extremely nice, thoughtful, kind, all the things that you can think of. But I wanted to know how this shy person got to know this woman. I mean, there is no chance that Govind asked for a date. So I asked Esther that one day, I said, how did you meet Govind? Man, the question nobody ever asked her. She lit up and she said, I saw this very handsome guy walking around in a youth festival, Budapest, Hungary. So I went to him, introduced myself, and spent all that day with him. And she said, I found him absolutely fascinating, interesting, as shy as he was. And so she said, we exchanged our addresses, and ever since we kept in touch, and here we are together. They were madly in love, believe me. And she took care of everything you could imagine. And she kept track of every one of us. And even the children, their wives, and every Christmas time, there was a card coming from Esther to everybody. I still have that last card. So, and she came to every Corona meetings, even when she had cancer, even when she was in a wheelchair. Total dedication there. I think I'm going to stop here. I have maybe someday at a different time, a lot of interesting things. Such fascinating personality, such a great person to grace this world. And so I'm going to stop here because we have people who are, there's a program next one. So I'm going to stop here. I already took extra 5 minutes. Oh, you can see that.
Thank you, sir. It was really very emotional and a wonderful memory. I request to our chairman to please continue 2 more lectures as we are running short of time and our speaker has to leave early. So I request to sir, please continue 2 more lectures.
Oh, I'm going to
as be a chair.
Oh, all right. Thank you. But I don't have the. Oh, Michelle. Well, Michelle, it's yeah.
So there's some confusion. I'll request Dr. Michelle on to.
Oh,
yeah.
Michelle, you are the chair.
Sorry about the confusion. Dr. Ranu has actually taken us to the whole new world, so there was that confusion. So we are starting, we are not going to have that tea break, and in the interest of time, we are starting with the next session, which is on plant biology, which will be chaired by Professor Michelle Lovin. I'll just briefly introduce you. So Professor Lovin has done her PhD in biochemistry from Queen's University. She was postdoc in University of Basel, and then she did her postdoc again from MIT, and she is presently adjunct assistant professor in Queen's University. I request her to please conduct the session. Yeah.
Okay, thank you. Okay, our next speaker in this wonderful field of plant biology will be Dr. Nagendra Singh.
Just mention his area of interest.
Okay, his areas of interest are genome sequencing and annotation of crop plants and microbes important for Indian agriculture, and provides support to plant breeders for molecular marker-assisted breeding. He's been the recipient of a number of awards, including the Queen Elizabeth II Award in 1988, National Biosciences Award from the Government of India in 2002, the Rafi Ahmed Award from ICAR in 2007, and the list goes on. And with that, I will pass it to Dr. Singh.
Thank you very much, Mr. Chair, and very good afternoon to each one of you. At the outset, I'd like to express my gratitude to the organizers for calling me here for this very, very interesting seminar, a symposium in the memory of Dr. Hargobind Khurana. Of course, we have grown up when we went to school. We just we heard about Dr. Khurana, as somebody said, when he won this Nobel Prize. That was the time when we were going to schools, to matriculation. And it was very inspiring to know that someone from India, after C.V. Raman, he was the one who got the Nobel Prize. Of course, after that, so many other people also got. So that is kind of inspirational personality he has been for all of us. What I'm going to, since time is very short, in this presentation I'm going to give a brief overview of some of the work which we have been doing in India under ICR about the genome decoding. And this is the outline of my presentation. Why to decode genomes? Of course, many of you know that. But I will just go through that, our starting initial decoding of some of the genomes of rice, tomato, and wheat through international partnership, and then one or two applications of that. Then we have taken up 3 different genomes, pigeon pea, jute, and mango. And these are the crops which are uniquely important for India with very high economic significance. But it's important for India mainly. So we have taken up these things and then how we have proceeded in that, and I will just summarize the progress in that. Of course, this genome word was coined by a German scientist called Hans Winkler at the time when we didn't even know that DNA. But his definition is still 100% accurate. It is the complete set of genetic information present in a haploid cell of an organism, including its protoplasm. So this is the same definition. It is true, and it is very complete and accurate. Why we have to do it? Because many people question, particularly in a country, developing country like India, with lot of resources limitations, that why you should spend so much money. It's very expensive to do genome sequencing. I have tried to put in just 4 points. It has applications right from the beginning for very basic application to the actual practical utilization in the translational research. The very first thing, when we have the complete physical map of the genome, of course Dr. Khurana was one of the team who cracked the genetic code, and he was the first one to synthesize the gene. And now we have come such a long way that we can have a complete map, genetic map of the chromosomes from end to end. And once we have that, then we can do a lot of interesting things through comparative analysis, compare different genomes and see that how these different species on Earth are they related to each other, and how they have the common origin, how far back they have the common parents. All those kinds of new questions, the Darwinian evolution, those can be answered. So this is a basic curiosity of all of us, that we want to know where we have come from and how we will be going. So it provides very interesting. We don't have time, but one of my students have developed an eukaryotic tree of life, taking about 100 genes which are common in all the eukaryotes, and you get very good, nice, beautiful tree of life. The second one, once you come down, you can use this genome information for characterization of germplasm resources. Now, germplasm resources are very, very important in the age of intellectual property right. We have been talking about this IPR, WTO, these kind of things. The nations or the countries or the people who have the intellect, they are very high in technology. They have that is knowledge, that is called wealth. So now we are living in a world where the brain waves are also protected as a wealth. But the vast majority of people in Asia and Africa and South America, which are very poor, they don't have any intellectual property. Only property they have is their genetic resources, or genome or the germplasm. So there is a biodiversity convention also to protect that. But how would you protect when you don't even know that you cannot fingerprint it? So once you have a genome information, you can have very high density fingerprint, and you can protect your germplasm resources. If any right you have, you can claim and argue for that. Then further down the lane, you can actually use this genome information to find useful genes from the germplasm. Because anybody will use that germplasm, only purpose is to get the genes. Because the genes are there in the germplasm, the resources. So that is really actually property which becomes really intellectual property, which can be used for useful purpose. So those genes can be mined. Having a genome information is just like having these GPS maps nowadays. These taxi people have, they can go anywhere. So having the genome helps to navigate through the genome to find the gene. And actually, last but not least, the actual breeding process as a plant breeders, when we do this selection, that selection process itself is helped by having the genome information. One can use the genetic marker for the foreground selection of the useful gene and the background. So these are starting right from the very, very basic to the applied. Any amount of money is spent on a genome which is important, a species which is very important for the economy, you get many times more in return. Now, this is a little bit complicated, but I won't go through here in more detail. It's just that we have different phases. You cannot complete a genome in 1 year or even 2 years. Usually they take quite a long time. Of course, even with a lot of development in the technology, still it is a slow process. Of course, it has become affordable and cheaper. So there are many species are there where we don't have any genome information. A lot of horticultural crops, fruit and vegetables, medicinal, which are very, very important, but we don't have any information. Then we can go to the preliminary draft stage, where 60 to 70% genome coverage. Many of the genomes have been published just like that, 60 to 80% coverage. Then few genomes which have the advanced draft, but not the reference level, just like a tomato, sorghum, chickpea, etc. Then very few genomes which we have very high quality draft, high quality reference sequence like Arabidopsis, rice or maize. Of course this number is increasing, but it's still the genomes the higher plant genomes you can count on the finger how many have been sequenced. And the reason why it is so difficult, because of the size of those genomes. If you see that the microbes have got only about 5 MB size, and now there is a technique you can sequence within a week. But as you go, see the wheat here, which has got 17 GB, which is 40 times bigger than the rice genome. So this enormous size of the genome gives a very challenging task to assemble it, because we have to sequence small bit by bit and then assemble back it like a jigsaw puzzle. So if the jigsaw picture is very large and pieces are small, it is going to be very difficult. And that's why wheat, even arguably the most important crops of the world, with the resource-rich countries, still it is not sequenced finished. It's only in the draft stage. We don't have a reference genome of wheat. India first started its genome journey through the rice genome project. Of course, we were not partner in the Arabidopsis and human genome project, so that way we were lagging behind at least 5 years through the international community, but we eventually joined in year 2000. Our center at ICR and Delhi University, Professor Khurana is here, Professor Tyagi was leading this whole program, and we took this chromosome 11 long arm. And this was a wonderful partnership. We got to learn so many things, and it was a really complementary partnership. And this sequence was launched in 2002 and took another 3 years to get it the finished quality. And it was published in Nature here. And this paper has been quoted more than 2,000 times actually. Although there were 2 more papers which were really preliminary drafts. And they were done in 2002, but this was published in 2005. But this is one of the most used plant genome paper for people who are working in this area. Now that sequencing was done, approach was clone by clone approach. We had to clone the DNA to sequence. Whether you have the 2-step cloning, like a large fragment clone, then a small fragment, or directly go to a small fragment whole genome shotgun. But cloning was necessary because you have to use the Sanger methods of sequencing. So it was used in the plasmid. And then with our experience, we got the responsibility to sequence chromosome 5 of tomato, because by this time we had learned all the tricks of the trade, we established the laboratories. I won't go into the detail, but it was quite expensive, and that's why in the beginning, in the tomato genome, which another had the consortium of 10 countries, it was decided to sequence only 20% of the genome, only the gene-rich region and the euchromatin on each of these chromosomes, 12 chromosomes. And then one has to find those bags belonging to specific chromosomes. And as it turned out that while sequencing, we find that the bags are going all over the chromosomes. Bags belonging to one country go to another country. And then in 2008, this next generation sequencing technologies came, the 3 independent technology. So this old method of sequencing, clone by clone sequencing, ended in about 2008. And it was found very slow and costly. This paper was published. And then in 2008, 3 new techniques came, 454, Illumina, and Solid, which avoided the cloning. We didn't have to clone the DNA for sequencing, because cloning was replaced by PCR. And as a result, it became much cheaper and much faster. So eventually we ended up sequencing about 90% of the tomato genome. And this was also a time when it became unnecessary to make a big consortium of the countries, because even a single country could sequence a big genome, because there was no need to have big partnerships. And it was not feasible also, because how will you divide the genome into many pieces? This paper was published in Nature in 2012, and in each of these 2 papers, in rice genome and tomato genome, there were 25 authors each from India. So our contribution was really quite intensive, because it was slow and steady things, and a lot of things, effort was required to reach that high level of standard to satisfy the international community. Wheat, I was just telling you that wheat is still not finished, and it is still possible to collaborate, because wheat is perhaps one of the rare plant species where you can separate each of the chromosome arms. We have a special kind of material called double dietylocentric, which was developed by the Ernest Sears in University of Missouri. For each of the 21 chromosomes, they are in 2 pieces. So you can do the flow sorting, and telocentrics are much smaller than the other chromosomes. So each of the chromosome halves were purified, and now we have chromosome arm specific DNA and the back library, which the 16 countries around the globe they shared. And then still they published this draft, which is only 60% of the coverage, couple of years back. So we are still waiting for the reference genome of wheat, but now we are into next generation of sequencing age. And then go to the next step. I will just give you one example here. The purpose of the genome sequencing is to do gene discovery, which can be used in for translational research to develop better varieties of crops, which can give you the higher yield or disease resistance or quality. So we picked up these three particular traits through forward genetics approach, assisted by genome information. This is this variety is a Jaya, the first one, which is the green revolution variety. Then we have the new material, which has got 400 grains per panicule, but they are not a variety, they are just a genetic material. We wanted to know what the gene which makes it happen. then special basmati quality and salt tolerance material. We took these three, and I will just give you only one example. This high grain number per panicule, we identified a QTL, a major QTL on the long arm of chromosome 4, and then by combining the different gene expression profiling, gene expression profiling methodology and genetic mapping, we narrowed down to just few genes in that region which are responsible for this high grain number. And at the same time, we mobilized this QTL into 12 different backgrounds. They are the very highly popular varieties of rice, and in each of the 12 variety, the tiller number was reduced, but the grain number per panicule was increased. And we have done the yield trial also. The yield also increases because of these. Most of these grains are actually fertile. They are well-filled grains. Now, with the same information, we have in our center, we designed a gene chip for genotyping on the Affimatics platform using the single copy genes. This was a special kind of approach which gave a very good insights into the evolution of the and domestication of the rice. And we found that some of the Indian rice varieties, groups like Indica rice and Aus rice, they have the closest wild rice relatives already present within India. So that showed that the rice has a polyphyletic origin. The Japonica type perhaps originated in the Southeast Asia or South China, but the Indica type and Aus type that originated within South Asia because they have the closest relatives there. And some of these, because there was a lot of controversy about this monophyletic or polyphyletic origins, and we found that there are mutations. The key domestication genes like the Saturning gene and red pericarp, and there are half a dozen genes which are very key to domestication. And it was claimed that there is a single origin of domestication because the same allele is present in most of the variety. But we found that there are other alternate mutations which also give the white pericarp and which are present in the Aus type of variety. So that was possible because of this high density chip which we had, gene-based chip, and there we could work out the haplotype. For each gene we had the multiple SNPs, and you can see that how many alleles are for that gene, and you can draw this relationship. Also we can use that chip for background selection. Like here is one of the examples of the variety I showed you. We have transferred this QTL here on the long arm. So this increases the yield by about 10 to 15%, and we recover rest of the basmati background, which is the blue here. So that means you maintain the original quality, everything of that variety, and you can transfer just the desired gene, just like you can do any of those genes. You can also do it for the bulk segregant analysis and identify the QTL, very much similar to what you will do when you use the biparental mapping population. But this study actually, we worked out some of the methodology related issues, that the original population should be very large, and the number of lines you bulk, that also should be large enough, then only your results should be accurate. Now let us come to the main thing which the title says, that the genomes which are economically important for India. So we took up this Pigeon P. We developed a consortium of Indian laboratories with in partnership with ICRISAT under Indo-US AKI, but as expected, there was a very less interest from the US side because Pigeon P is not important for US. So, but we developed this thing because see, India is the largest producer and more than 90% is produced and consumed in India. So Indian government put this money, and at that time, by this time we had learned how to use the next generation sequencing technology, and particularly this 454, which was easier to assemble through bioinformatics, and we published the first draft of the Pigeon P genome in 2011. This was the first complete genome done entirely within India. And similarly, in Delhi University also did the chickpea for that, because chickpea is similar way, it is very, very important for India. So once we learn through international cooperation, then one can use those techniques for the translational purpose within the country. So then Pigeon P was such a craft that there was absolutely no information available there. So here generating these resources